Hello and welcome to another National Insect Week 2014 podcast. I'm joined today by Professor Lin Field of Rothamsted Research. She's worked extensively with the chemical basis of controlling insect pests. So thank you, Lin, for joining us today.
You're very welcome. Welcome to Rothamsted.
Thank you very much. So, first of all, how important are insects as pests and vectors of diseases?
They are very important. Well, in two respects they're very important. One is vectoring human and animal disease. Mosquitoes vectoring dengue or malaria. And in the crop world, the big problems in Europe are largely aphids transmitting viral diseases. But worldwide, there are all sorts of other insects where the larvae eat the crop and do direct damage, so cotton bollworm or things that eat maize. There's a whole range. So they are very important.
Insects as pests was a big problem. Chemical development was a big area that needed to be tackled.
And it still is a problem in that there are very few new groups of chemicals that come on the market, and predictably, at some point, a chemical fails because of resistance. This is the same as antibiotic resistance in a hospital. Because you have lots of bacteria, you get resistant. You have lots of insects, you get resistance.
It's not just a case of applying a chemical, it works, it kills most things, your crop yields increase. You can't just keep using it for all time.
Well, there are two issues in what you just said. You don't want to kill everything. That's the point. So at one point when people were designing insecticides, the old insecticides that came onto the market, they were tested for toxicity to mammals, and there had to be a difference in that they had to kill insects and not mammals. But more and more now we want insecticides that kill some insects and not other insects. So we're looking for specificity of killing, say, a mosquito in Africa, not killing a beneficial insect, or killing an aphid on a crop, but not killing a ladybird or a bee. So it's getting much more sophisticated now, the kind of chemical attack on insects, because we don't want to indiscriminately kill everything. I have noticed a change in attitudes with companies. Companies are much more interested now in getting chemistry that will last, won't have the resistance problem, but also will only kill the insect they're after. And that might be wanting to kill, say, a varroa mite in a beehive and not kill the bee. It's quite a challenge, and companies are rising to it. I find it exciting because I think it is opening up better control with less toxic chemistry around.
So how do insects develop resistance?
Okay. Well, there are various mechanisms. So when you first start using an insecticide, it's killing the insect by acting on a particular protein in the insect. Now, the insect has some options here in terms of evolution. It can evolve a system where it is more able to detoxify the insecticide. So the insecticide never gets to the target protein. So this might be an increase in a particular group of enzymes. It's usually esterases or cytochrome P450s. We all have these kind of defence enzymes that detoxify toxins. It's the reason why we can eat certain things that could be toxic in theory, but we detoxify them. And insects have this, and by increasing the amount of enzyme, they can increase it to a level where they can detoxify the pesticide enough for it not to get to the target at all. That's quite a common mechanism. The other main mechanism is that the protein that the insecticide has to interact with actually changes. So there's a mutation in the gene that changes the protein, so the protein still functions, but the insecticide doesn't bind. We call that target site resistance. That's a very specific DNA mutation. All these mutations are arising by chance, obviously, and then being selected. So it depends a bit on which mutation arises and then subsequently gets selected. Because once it's there, it's such an advantage. Because the insect's not dying. You can't get much better, higher selection than that.
So this is when you don't kill everything, and the ones that are remaining have this adaptation.
Exactly, and then they pass that on to their offspring. And sometimes that passing on is incredibly successful, because in aphids, for instance, which are one of the main crop pests, they reproduce largely asexually. So once you have a mutation, it doesn't even go through a sexual stage, it just gets passed on to all the offspring and all her offspring.
So it can be really rapid.
It can move very quickly.
So the insecticides, they target a protein. What kind of proteins are they targeting?
Okay, so most of the main groups target proteins in the nervous system. So the pyrethroids target a thing called the sodium channel, which is a protein in the nervous system that allows sodium to move in and out and creates the action potential in the nerve. Now, we all have sodium channels, mammals have sodium channels, but pyrethroids are a very, very low toxicity to mammals, and that's because mammals metabolize them very effectively. And actually, we know now from our work that mammals have a slightly different sodium channel anyway. The sequence of the gene is slightly different, and that prevents pyrethroids binding to the mammalian channel, but they do bind to the insect channel. And when you get resistance, you get a mutation in the sodium channel gene that stops that happening. The newest group, the neonicotinoids, which are quite controversial, they target the nicotinic acetylcholine receptor, which is again a receptor that allows neurotransmission. But again, the mammalian receptor is very different to the insect receptor, and they bind very, very low levels to the mammalian receptor, which is why they've got good specificity for insects and not mammals. But again, what we really would like is one that differentiated between the nicotinic receptor of one insect and not another. And some of the work we do here is trying to, especially now we have insect genomes, is looking at subtle differences between these proteins to try and find ideas for designing a compound that would kill an aphid and not a bee.
So this is where genetic methods and really understanding the cellular structure of the organisms involved.
It's where first of all, it's where genome sequences are becoming very important. So we're now able to sequence the whole genome of an organism. The first insect we ever had was Drosophila, which of course is a model insect. It's not a. But now we have lots of pest insect genomes. So we have 2 aphid genomes now. We have various other genomes that we can go into the database without doing any wet science. We can go in and we can find the gene that encodes the target protein, and we can look in detail at what it looks like. We can model the 3D shape and how it interacts with the pesticide and kind of really understand how the pesticide is actually working. And sometimes those models allow us to say, although you've got a mutation in your, say, sodium channel that will stop this pyrethroid binding, because a different pyrethroid is a slightly different shape, it probably will still bind. So you may have resistance to one pyrethroid but not another, and that kind of thing can then inform control strategies out in the field. Obviously, in the long run, we'd like designer insecticides in the same way as people work towards designer drugs, where we make a molecule that targets exactly what we want it to in the way we want it to, but that's still some way down the line. The big advantage now is that you don't have to assume a prior knowledge, in that we don't just go in and look for P450s, which we know give resistance, or a mutation in the target site. We can directly compare a sensitive insect with a resistant one and saying, what is it making? What is one making that the other one isn't? Is it making a new enzyme? Or is it making something that would change the cuticle? So it's giving us a much more objective view of what kind of mechanisms might crop up. And I think we will start finding some surprises, because we have not had that ability. So yeah, the availability of genome data is just amazing in my lifetime.
So at Rothamsted, how could you describe the role of your group and your work here in contributing?
My own group is insecticide resistance, because that's where I started, but the insecticide resistance work is going to carry on being important. I think in the foreseeable future that won't go down. If anything, I see it going up. But the other aspect of the department, which is trying to develop novel control strategies using semiochemistry, understanding how insects move around. Insects are incredibly good at responding to volatile chemical signals using olfaction. Very exquisitely tuned to find one plant or one animal host. I'm trying to understand that, trying to disrupt it, trying to use that in novel strategies is going to grow in importance. And that's a big area of this department's work, which I think will become increasingly more important.
So whole organism biology, studying how insects behave, is becoming really important.
That goes right through from understanding how an insect, what plant an insect is being attracted to. We then have a really neat system where we can collect the volatiles that come off the plant. That's always a mixture, a complex mixture. We can separate the individual components out on a gas chromatograph, pass them over an insect's antenna that's been wired up, and see exactly which compound the insect is identifying. Then we can go back to behaviour to say, is it identifying this compound as an attractant or a repellent? So that's quite sophisticated electrophysiology. And then we can go inside the antennae and say what proteins inside the antennae are responding to this signal, which proteins are binding to the volatile and triggering the behavioural response in the nervous system. So that area of science ranges from behavioural studies right through to gain cloning genes and understanding how they interact with these volatile chemicals.
So it's a real cascade of discoveries.
It is, and we have all that expertise in the department as a group, chemical ecology group, which does literally go out in the field and stick electrodes in antennae and clone genes. So yeah, we have all that coming along, which we hope certainly in the next midterm will start yielding new ideas for control.
You're also looking at the effects of I always stumble on
neonicotinoids.
Thank you very much.
Yes.
The study of neonicotinoids on bees' health, and this has been in the news. This is big.
Yes, it has been big
on the public conscience as well.
It has. Yes.
You've said farmers need these chemicals to protect their crops, but there is such a pressure to be ecologically friendly.
Indeed, yes.
Is there a way both of these can be married together?
Well, the irony is that I always thought, and a lot of people I think did, that neonicotinoids were much better than other pesticides because they were used as seed treatments, grew up with the crop, and therefore were directly affecting the pests landing on the crop. You weren't spraying them. So in many ways they were more friendly towards non-targets like bees. Then the question started to be asked, well, is there now a risk that some of this seed treatment is ending up in the pollen and the nectar, which bees are then collecting and which are being affected? Now, I have no doubt that neonicotinoids can kill bees. They are toxic to bees, as are pyrethroids or any of the alternatives. So the question really is it doing it in the field? Now, you can measure neonicotinoids in pollen and nectar at low levels. You can also show that at low levels, or some low levels, neonicotinoids can affect behaviour of bees without being toxic. And I'm not doubting either of those sets of data. What my question is that actually causal in the field? Are the bees picking up enough contaminated nectar and pollen to be seriously hampering? And I don't think we really know the answer to that. I mean, people have done experiments where they feed neonicotinoids to bees in the field through feeding rather than the nectar and pollen. But often the levels are much higher than you would expect them to be getting from nectar and pollen.
So you're saying they've done studies where bees are fed neonicotinoids
at sublethal levels, so they don't die, but it affects their behaviour.
And then you're suggesting that in the field they're not having a diet just of these neonicotinoids.
They may not be. In fact, there is some evidence, and again, I'm not sure how good it is, that bees are actually put off feeding on something contaminated with neonicotinoids. So it's quite hard to know. And bees, the crops that flower that bees go into, which in this country is mainly oilseed rape, they're only feeding on oilseed rape for a certain amount of time, and I'm, without knowing all the foraging behaviour, and I'm not a bee behaviour expert by any stretch of the imagination, it's quite hard to know how much they would pick up. You can certainly detect it back in hives, but whether that's actually causing bees to get disorientated, for instance, and not get back to the hive, there are some experiments trying to look at this. We've got some work here trying to do it, where we've got trackers on bees, but they're difficult to do, and I think. So whilst I would not sit here and tell you I'm absolutely certain neonics are not damaging bees, I'm not certain that they're the main cause. And I'm not certain that the ban that's now come in place will solve the problem, because clearly bees have all sorts of other problems. So they have this varroa mite that's spread around, the mite carries viruses, we've had some quite difficult weather conditions, the climate, Bees are facing a whole suite of issues, and if we think we've solved the whole problem by banning neonicotinoids on certain crops, I think in a way we do the bees a disservice, because we take our eye off the fact that there will be other issues out there. My own personal view is that the ban is not in itself going to solve the problem, but if it does, fine, but there's no real evidence that it will, because let's look at even if we look at correlative data, they have banned neonics in sunflowers in France for quite a lot of years, but I don't think bees have recovered, at least I haven't seen any evidence. And in Australia, they use neonics quite widely, but they don't seem to have bee problems so much, and that's largely because they've kept the varroa mite out. So even looked at correlations, it's hard to see. The data are very difficult to assess. You can kind of assess it either way. We know something, but we don't know enough to really know whether neonics are the main problem for bees.
So if you could convince the next generation of entomologists why coming into insecticide resistance research is important, why would you convince them this way?
At a real practical level, if you want to become a professional entomologist, you have to find somebody to pay you for what you do. And crop protection is an area where there is a possibility of getting a job. And so crop protection is something that, instinctively you might think as an entomologist, well, I'm not sure I want to really do this, because you're actually trying to kill an insect. But it is part of developing good systems for crop protection, crop production. So if you are passionate about the need to feed the world, and that sounds totally grand, but the need to produce food, then it is an area that is very rewarding, trying to understand and get better control of pests.
Well, Lynn, thank you very much for your insights into a completely different area of entomology that we've had so far. Okay. So thank you very much.
Thank you very much. I've enjoyed it.